This is a project that is yet again one of those things where I'm standing here and telling the story, and it's only because so many people helped that I have a story to tell. So the NASA developed team that we worked with, they have a number of centers throughout the country. Each semester for 10 weeks, they choose particular projects that are brought to them by community members. So we came to them saying, help. I also want to say thank you to Supervisor Sheila Kuehl. She provided the main support for this, augmented by the National Park Service, Southern California Research Learning Center. They also contributed funds to this, and the LA County Fish and Game Commission gave us the money for the lures, which was really incredible. But this whole project wouldn't have happened without this cast of characters who, whenever I come up with these brain-damaged ideas and say, gee, let's set out traps and go count trees, and they go, sure, let's go do it. And they come and they help. And it's really an amazing thing. This project alone had over 2,000 volunteer hours to make it happen. The U.S. Forest Service has done all this big work looking at the number of dead trees throughout the Sierra Nevada and other places, but nobody has looked at the Santa Monica Mountains, and part of the reason why is because we don't count as a forest. We don't have pine trees that are easy to count when you fly over them because they like stick their pointy tops up. And we also have a very density of our trees. So when looked at on a 10-kilometer grid, it doesn't look so exciting, but actually we have quite a lot. The study area is the Santa Monica Mountains National Recreation Area plus. So you can see that the boundary extends a little bit further north up into the Simi Hills, beyond the normal boundary of the National Recreation Area, and includes most of the western San Fernando Valley, our mountains, and parts of Ventura County. What's really important here is that this study area covers quite a lot of different kinds of plant assemblages. And there are over 400 associations of vegetation classification that National Park Service has mapped. And that's an important thing to remember because it explains how complicated it was for the NASA folks to actually take that data and make sense out of it. Why do we care about oak trees dying? Well, this is a crowd that I don't have to explain that much to, but oak trees are keystone species. They are the hub of the wheel of a lot of life. And in fact, there are over 5,000 species of insects, 58 species of reptiles and amphibians, 150 species of birds, and 105 species of mammals that would not be here in the Santa Monica Mountains if we did not have oak trees. So losing them is a big deal from an ecological perspective. But we also have, and what became really apparent in the course of this study, thanks to the NASA folks, was a lot of impacts to our riparian trees. And they hold an even more special, crucial part because the shade from those trees, the roots that hold the banks, the leaves that fall in to provide food for all the bugs, shade trees are critical for much of our aquatic life. And we have federally listed species like the steelhead trout and the red-legged frog. We have many species of special concern. All of these aquatic species are going to have a lot of trouble if our riparian forests disappear. So when you start a study, you ask questions. And really good studies generate more questions than they do answers, which of course is what happened here too. But the questions that we started out with are, why has this drought been so much more difficult for our vegetation to survive than previous droughts? And what are the synergistic relationships between drought and temperature and perhaps the numbers of trees and how we're managing our trees and our fire suppression and all of these things? And then add in these new introduced bad beetles, and you've got a mix of potential stressors. So the idea here was to sort this all out. It's really a story in many parts, and they all weave together. And what's, I think, important here is to recognize that it's never simple when you start trying to look at what's going on in the natural world and how we interact with it. So, part one is the drought. Don't have to tell you guys how bad the drought's been, but I think what's really scary is that we're really not out of the woods yet, so to speak, with the drought. And even though we had some nice rains last winter, it really has not been enough to revive the trees, revive the water table. It's pretty rough out there. Alex Hall, UCLA, has done some really interesting climate modeling. And he has, put together a few slides about how temperature has changed in LA. And although 6 degrees Fahrenheit doesn't sound like a big deal over not quite 200-year period, it's a really big deal if you're a tree. And you're a long-lived vegetation species that's stuck in one place, and you have to deal with whatever comes your way. And you can't move, you can't run away, you are where you are. And so changing the temperature regime like this is a really big deal. So our colleagues at NASA did an amazing job of pulling together a huge data set based on 4 kilometers, so that's about 2.5 meters square. So every one of those boxes that you see in the slide is kind of a 4 kilometer square. And they averaged together all the temperatures to get the mean temperature. And so what you notice is that the eastern side of the Santa Monica's is darker color, which means it's hotter. And you can see the progression of change over time. This one is the data's only till October, so it's not quite as detailed as the other maps. But you can see that temperature during the drought was definitely an issue. But what's really interesting is that people get pretty unhappy when days are over 95 degrees. Well, it turns out oak trees do too. If you look again at this band of days over 95, again it concentrates over in the eastern and northern part of the study area. Hold that thought for as we go forward. One of the hypotheses that we put forward is we said, all right, is it the number of days, or is it the distribution of those extreme temperature days? Are there more of them just at one time of year, or are they spreading themselves out? And so Brianna, who is back there, did a great job looking at the data from Woodland Hills, from Pierce College, and from Malibu Hills, which is a station at Las Flores, right near the fire camp there. And she put together this slide, which is not meant to be looked at with great detail, but if you look, the red is Woodland Hills, the blue is Malibu, the purple is Leo Carrillo, which you can barely see, because Leo Carrillo doesn't get very many days over 95 degrees. But if you look at the red, you can see that during the past couple years, things have been pretty intense. And the concentration of hot days is July, August, and September. But this doesn't really quite tell the whole story. And I think this is one of the things that we really have to delve into a little bit more, because this doesn't necessarily match this same image here. And so I think it's not because the data is incorrect. I think it's because we haven't figured out how the story needs to be told yet. So stay tuned on that. But what's really scary is Alex used a bunch of climate models, and he said, what is the projected number of days over 95 degrees in 2050? And that's the middle bar. And if you look at the Los Angeles one, it says 22, and we're already at 44. So at 2017, we're at 44. We haven't even made it to, I mean, we're way ahead of the predictions of this model. So that kind of sets the stage to looking at what Megan Williams started, with emails and questions years ago in 2015 and said, Rosie, why are all the oak trees in Topanga State Park dying? And so we realized we really needed a baseline so that we could look and see whether trees were dying, how many, where, what was going on. And this map from the Oak Woodlands Conservation Management Plan kind of gives you the pink blobs or the distributions of oak woodlands. And you can see that a lot of them follow riparian corridors. right? They're following the creek corridors. And so one of the big questions we had then is this just happening up in the uplands, like at Topanga State Park, up at Trippett Ranch, or is this happening in the riparian corridors as well? Where is this all going on? So with the help of Denise Camrat and some of her colleagues at National Park Service, they very graciously generated this grid map for us of 25-meter plots. And Jen Mongolo, who's here, also helped with setting this whole thing up. And we gave them random numbers, and we randomly pulled plots that we could then go in and tag every tree in the plot and assess their health and condition. We now have 41 plots throughout the Santa Monica Mountains, 353 trees tagged, and this gives us a baseline that we can now start to follow over time. And it's been really interesting to see the distribution. We specifically selected areas in riparian corridors where it was either adjacent to or important habitat for either red-legged frogs or steelhead trout, because those were our species of concern. And what we learned is something that everyone who goes hiking in the Santa Monica Mountains probably has kind of already felt, but never really put their finger on, and that is that we have a very aging forest. And most of our trees are middle-aged to old. That makes sense because up until 1915, any oak tree that was easily accessible was cut down for firewood because oak was the fuel at that time. And it wasn't until the early 1900s that electricity became available and oaks were not cut down as much. So we're seeing a huge regrowth since the early 1900s, and that corresponds with fire suppression. And not necessarily letting small fires burn. We wait for the really big ones, like tonight. And so this combination has changed the density of trees per plot and per area. And that could be part of the story. We don't really know. But it kind of points us in the direction that we probably really have to be thinking long-term here and get our act together to start planting trees. But more about that later. So we started looking at specifics, and the trees that were in plots that were more densely populated were slightly less healthy condition. And what's really staggering is that when we started in 2013, only 1% of the trees were dead, and by 2017, over 20% were dead. We also tried to look at correlations between the location of the trees, whether they were north-facing slopes, south-facing slopes, whether they were high up on the slope, lower down, whether the slopes were higher or lower gradient. And there weren't a whole lot of significant correlations there, but definitely the south side got hit harder. And that kind of got us to the point where, okay, we can look at these individual trees in these individual sites, but that only tells us about that site. And really what we wanted to know was what's happening in the entire Santa Monica Mountains. And that's a landscape level plan. And so with that, I will turn it over to the NASA folks because they will be able to explain what they did on that. So come on up, gals, and we'll introduce you all.
All right, so Rosie talked briefly about what the NASA Develop program is, but I just wanted to give a little bit more detail about it. So the NASA Develop program is a part of the Applied Sciences Division within NASA, and what it does is it aims to get recent college graduates in a team and partner them with a science advisor who collaborate together over a 10-week period to address a community concern of some type using remote sensing techniques. And so the title of our project is Utilizing NASA Earth Observations to Determine Drought, Dieback, and Insect-Related Damage in the Santa Monica Mountains, California.
So at the beginning of our term in September, we met up with Rosie and some of the other members of the RCD and TAC team, and we decided what our objectives were going to be. And so from our shared interests, we determined that we wanted to look at how much of the green vegetation of different species had been lost over the course of this drought in our study area. And we also along with climatic variables, wanted to look at the topographical variables of the mountains, such as aspect and elevation and slope, and see how that was affecting dieback. And finally, identify areas that Rosie and her team could look at so that they could focus on areas that are probably more susceptible to drought in the future. The satellites and sensors that we used were the SRTM, the Shuttle Radar Topography Mission, which gives us images that we can use to look at elevation of the mountains in pretty high detail, and AVIRIS, which is an airborne sensor, which lets us look at the spectra of the vegetation in much higher detail than most other sensors, and we can begin to parse what vegetation is where.
Okay, so because of our objectives, we wanted to do a couple things with the AVIRIS. First of all, we wanted to see where trees were dying, which meant we had to figure out some way to map plant mortality. And we also wanted to see specifically how this was affecting oak trees, so we would want to discern species in some way. So for both of these goals, we used a method called multiple end-member spectral mixture analysis, or MESMA, as I'll call it from now on. It classifies your image based on spectral classes that you give it. So you can create your training data using a spectral library. A spectral library is just how a certain class reflects across the electromagnetic spectrum. And once you've defined your classes, then you can classify the image with your spectral library. So to try and assess plant mortality, we had 3 classes: green vegetation, non-photosynthetic vegetation, and substrate, which would just be dirt. This is what the spectral library looks like. You can tell just the way the different land classes reflect. So substrate can be seen in blue, and that sort of arches over at the top. And the green vegetation is in green at the bottom, and it has this very distinctive, what they call the red shift. So the shift from red to near infrared, that would be, you can see the green goes down and then way up. And then non-photosynthetic vegetation is in the red in the middle. And for each pixel, MESMA outputs fractional cover for each of those classes. So for each pixel, it would say it's 50% green and 40% non-photosynthetic and 10% substrate, something like that. And so we wanted to see how the green vegetation and non-photosynthetic vegetation was relating within each pixel. So we calculated relative fraction of alive cover, which is just green vegetation over all vegetation, green plus non-photosynthetic. So for each pixel, a little model here, those squares, each square is a pixel. For each a pixel, we would have a value of the relative fraction of alive cover. So something like a 0.6 or 0.8, that's quite high, 60 or 80%. And we knew that things lower than a certain amount were low, which would mean mostly dead. This is just our classified image, right? And we wanted to be able to relate it to what we already knew on the ground from what Rosie was doing with the oak plots. And so to do that, we wanted to find some sort of threshold that could relate what our relative fraction of alive cover meant and their tree conditions meant.
Yeah, so in order to do this, we overlaid our relative fraction of our alive vegetation map with the oak plots that Rosie has. And once we did that, we wanted to figure out what the average of our relative fraction of alive vegetation would be per each plot. So what the end result would be an average RFAL value and a value of the plot condition. We then used those values to see what the relationship between the plot condition was and our RFAL values in order to find a threshold from which we could say everything below this number is dead and everything above this number is alive.
So once we were done with finding this threshold where we felt more certain that below certain RFAL number, our pixels were dead, we wanted to relate those to the different species covers that we had. And so we wanted to make a species map, and this is our basic outline of the methodology. First, like I showed you before, I wanted to build a spectral library, but instead of focusing on green vegetation, non-photosynthetic vegetation, I had all of the different classes of vegetation types that we had worked out with the partners that we wanted to study. So that would include chaparral, coastal sage scrub, riparian, oak woodland, annual grass, stuff like that. Once we had our spectral library, we would use that same method from before, MESMA, to map out the vegetation types across the landscape. We had the dead pixels located from doing the thresholding that Kelsey talked about, and then we could see where dead pixels were intersecting with certain vegetation types. And once we had those numbers, we could calculate acreage lost. So to make the spectral library, what I would do was I would use the NPS vegetation map that we had been provided, and I would look at a certain class type, and I would compare it with really high spatial resolution imagery to see if it looked like that vegetation type was actually there, just making sure. So here you can see in blue, that's an outline of an oak woodland area that we got from the NPS vegetation map. And then as you can see in the imagery, it does appear that in fact there are oaks there. So I would just I would make my own little polygons in there, as you can see in red, where the vegetation seemed very dense, because when you're making the spectral library, you want it to be the most pure representation possible of the class you're looking at. So I'd pick dense areas where it didn't seem like there was much dirt or other vegetation types showing. And then I would overlay our actual avarice imagery, which is what we use for the classification, because you want to make sure that not only is it lining up, but the pixels you're choosing aren't actually on the edge of the polygon and mixing with a different type. So once I had the avarice imagery overlaid, I would pick all of the pixels that were contained within the polygons I had chosen. And so from then on, you can extract the spectral reflectance from each pixel and then build your library from that. So as you can imagine, doing that method, I had over 2,000 spectra, which is way too much. You don't want to be running models with that much data. So this is what the library looked like pared down using a certain method that's built into Viper Tools, which is what I was using for this classification. And this is lowest, or n-member average root mean square error. And so n-members are each of the spectra that I had extracted, and essentially you can use this EAR, n-member average root mean square error, to unmix certain classes with each other and see which ones are the best representations. So some of them might be too similar to each other, some of them might be too different. So getting the 20 lowest EAR for each of those, that's what you see here. But even with that, I could tell that there were some outliers that didn't really make sense, which is just mostly because I'm familiar with what the spectra looks like. But for example, you can see the substrates in brown, and although it typically arches along the top, there are a couple that seem to be mixed in with the vegetation lower below. So stuff like that I would take out manually. And so that would give me this. And then from there, all I did was divide them in half. Each class, I would divide the spectra in half randomly, so that one half would be used for the training and the other half was used for validation. So we could make sure that what we were classifying actually was accurate. Here you can see the final result. This is what the species map looked like. These are the classes we decided to go to. We had annual grass, Ceanothus megacarpus, which is big pod Ceanothus. That's a chaparral type that our partners told us was very affected by the drought. Ceanothus spinosus, that's another chaparral type. Chaparral common, which included a few other things, scrub oak, chamise, I think, and one other species. We had two classes of coastal sage scrub, one that tends to be more drought deciduous, one that tends to stay active in the summer. Then we had Melos melorina, which is the laurel sumac, coast live oak woodland, riparian, substrate, and water. You can definitely see certain trends. Riparian goes along the stream beds. Annual grass definitely covers the area where there was a large fire in 2013, and so on. But after I assessed the accuracy of this map, I did see that trying to get down to the more specific types, so to say, there was not as high accuracy discerning between different chaparrals or different coastal sage scrubs. So I also just made a map that merged the chaparral and the coastal sage types, and you can see that here.
This is jumping back to more of our second objective, which was to try and see what how the physical constraints were affecting dieback. And so on the first column in blue, you can see the number of days with precipitation from 2013 to 2016. In the middle is the number of days over 95, and on the furthest side is the dead vegetation. And so this is classified based off of that threshold that we came up with, which ended up being about 0.5 of our RFAL value. So anything below 0.5 was what we decided to classify as dead. One thing to point out is that you can see that there's a trend in where there is dead vegetation and where there were extreme heat days and where there was lack of precipitation. And it all seems to play together into a story like Rosie was mentioning of how this landscape was affected by the drought. So using the species map that Natalie made and using our thresholds that we found, we were able to determine the acres of alive vegetation for each of the species types that we had in 2013, and also the amount of acreage that was lost by 2016. Most important, I think, to point out is that we lost about 9,000 oak woodland trees.
So we also have the opportunity to work with LiDAR data, which stands for light detection and ranging, and is another form of airborne sensing, which creates these things called point clouds, which can give us 3D representations of the surface that the sensor is flying over. So this is a cross-section of, I believe, an area of Trippett Ranch, and the different colors are showing different, basically, elevations of the point cloud. But if we want to sort of make that more friendly to what we were working with, we can create a digital surface model, which is taking the points that are sort of, I guess, the highest of each area, and a digital elevation model, which is solely the terrain. And the difference between those is going to give us our canopy height model. So then we can just have the points for the heights of the trees. And here is the area around Trippett Ranch coded for canopy height for that. And we had this idea that maybe we could use this LiDAR data to look at areas where there were increased fire danger. So using our DSMs and DEMs, we were able to find the density of the trees in different areas and multiply that by the 1 minus our RFAL, which gives us the fraction dead, which was going to give us the amount of dead fuel that was around and going to be potentially very dangerous in times of fire. So the areas in red, as you can see, are areas of higher fire danger, and the map on the very left is what you would see if you were to zoom out pretty far in the Santa Monica Mountains, and you can see you can't see very much, but LiDAR data is so fine that when you zoom in, you can really see a lot in terms of where we should be looking out for danger like today. We, of course, had errors and uncertainties in our project. For instance, there was a temporal discrepancy between the time Rosie and her team collected the field plot condition data for the trees in the fall and when the Avarice sensor flew in June and May. So there could be some discrepancies there because, as we know, the trees have been dying over very short time spans, and there could be some uncertainties there. Additionally, the spring imagery of the Avarice may be influenced by the phenology. And as Natalie was showing you, individual species can be pretty well mapped using MESMA, but there's always room for improvement. And finally, sort of brushing on what Rosie was talking about earlier with looking at how days over 95 are dispersed or numbers like that, we may be just looking at things with the wrong temporal window, and this misalignment may be causing us to miss some of the trends that we otherwise would see.
So I just want you to see, from that really incredible, high-tech, amazing stuff, we go to the homemade detection detective. These were our bad beetle traps. And, nothing like a little DIY to get things going. But I have to credit Dr. Richard Stotthammer at UC Riverside. He was the one who came up with this really cool 3D thing that the public library in Thousand Oaks printed out for us in a 3D printer that connects the bottles. So that was pretty spiffy. What we wanted to know is, now that we had kind of this bigger picture of we've got these plots, the NASA team was able to look at it on a landscape level in terms of the drought and the temperature. The next question we had is, what's happening with these bad beetles, and where do they fit into this, and what role are they playing in causing this dieback or exacerbating it? So we have all these native insects that are associated with both our native oak woodlands and our riparian woodlands. And these trees have lived with these bugs for thousands of years, and nobody has had excessive dieback on a regular basis. But now some of our native bugs, like the western oak bark beetle, are carrying new fungi that is different from what they've had before. And so now, instead of killing dead and dying trees and being sort of a secondary pathogen, the western oak bark beetle is now carrying a fungi that, like these other insects that we're going to talk about, is its own pathogen. So now the trees are getting hit 2 times. It's a double whammy. First, the problem is that the beetles are boring in and mechanically, physically digging tunnels that disrupt the movement of water and nutrients through a tree. But secondarily, all of those tubes are now filled with fungi that are killing the trees. And it's not really clear which does the worst fastest. They're both pretty aggressive. And that's what's new and different. So the foamy canker disease is really a key feature in what's killed most of the trees in Trippett Ranch. What's fascinating is that we have these other new bad actors. The gold-spotted oak borer is native to Mexico and Arizona and got transported in firewood in about 2004, I think, to San Diego County. They lost 80,000 trees in 4 years to this bug. It has recently been found and confirmed in Santa Clarita and Green Valley. The LA County Fire Department has removed over 100 trees. They've identified 400 more. And these bugs are bad, 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 bad. But they only go for oaks. The really scary guy is the Polyphagus shothole borer, who's now been renamed the invasive shothole borer. They keep changing the names. But the problem with these guys is the same. I just wanted to show this one slide. You see the hand holding that little piece of bark? That's like 1 palm's worth of bark. That one piece came up with 186 beetles. So like the shothole borer, like the western oak bark beetle, these things reproduce madly and very successfully. And they do it underneath the bark so that they're very well protected from any kind of predators. The shothole borers, however, don't just go for oaks. There are a huge number of trees that are now reproductive hosts. I think it's 75 species are now reproductive hosts. There are over 300 species that have been attacked by this beetle. So this is the one that could be the total game changer in terms of our landscape in Southern California. And then, because life is never dull, down in the creek, we found this grove of alders, which if you saw the LA Times article this morning, that's where I took the reporter. You know, we have this incredible alder grove, and in this one small reach of creek, and almost every tree is dead, and they all look like they have the measles. And it turns out that this is yet another introduced invasive beetle called the fruit tree pinhole borer, which also carries a pathogenic fungi. So these guys are all doing the same kind of thing, just different species carry different fungi, but the result is the same. So one of the things that we were able to do with our plots is to look at how many trees in each plot had evidence of boring activity from these beetles. And in 2015, when we started the plots, they were all native beetles, not bad beetles, some western oak bark beetles, but nothing else. By 2016, the western oak bark beetles had taken over. And by 2017, we now had the whole suite of characters except gold-spotted oak borer. We still do not have gold-spotted oak borer in the Santa Monica Mountains, and we really hope that it stays in Santa Clarita and doesn't come to visit. So, you know, now we've got these lists of plants that are all being hit by these various beetles. And one of the things that keeps coming up is, well, what do we do? What do we replace these trees that are dying with? Do we put back these trees, the same species that are being attacked, or do we use something else? Well, it turns out there's nothing else, because the Polyphicus shot-hole borer will go for anything, basically. So what I think that tells me is that our key to responding to this, one of the major things that we can each do, each one of us individually, is plant a native tree. Because the ripple effects of losing those trees is so significant that everything we can do to plant these trees is key. So this is just a scary map that shows the distribution over time of the shot-hole borers. It's really pretty crazy. And these little things, I'm going to, pass these around. Many of you have seen them, but they're the size of a sesame seed, of a grain of rice. They're tiny little beetles, and they can't fly very far. So the way they're getting moved is people are having trees that die. They have people come in and cut them down, and those guys transport the firewood and the slash from those trees. Most tree companies do not have chippers that can get material down to 1 inch or below, which is what it takes to control the spread of the beetles. So they leave the firewood, they chip it to a certain degree. We think that in Calabasas, the reason we've got so many infections is because of bad mulch. So it's a real problem, and this is why the riparian areas are so key to this whole story. Please note that this photo was taken in May of 2015. What you're not seeing is the backyard of the guy who got a load of free firewood from his buddy. And this is what it looked like not even a year later. This is the Tijuana River Valley down in San Diego. And every single willow, something like 140,000 trees within a mile, were completely decimated. And it happened in 6-7 months. It's really scary. And what's even scarier is that even as these willows regrow, because they will resprout from their base, what happens is they get to a certain size, and then the beetles come back and get them again. But we got to keep going, because if we lose all these riparian trees, we're definitely going to lose our species. And what does all this mean? This is Sal, who's sitting right here, and Sal deserves really a gold medal, because every week all of our amazing volunteers would bring in 45 or 50 little samples in coffee filters. And Sal would very painstakingly pull out every bug from these coffee filters and sort them into bad beetles and good beetles. And the bad beetles all got put into vials that were sent down to UC Riverside, and Dr. Stottheimer's lab actually did visual confirmation of them and some DNA to make sure that we had bad beetles. And the good beetles are all going to the Natural History Museum, and they're an amazing collection and resource. Somebody's master's thesis is waiting to be done. This is kind of the summary of what we did with the bad beetles. And Sal did over 850 samples in 27 weeks. That's pretty epic. That's a lot of microscope time. Yay, Sal. So of all of those samples, though, the good news is only 52 of them were bad beetles. And they were in the same 15 traps. So what we learned is that where they're bad, they're really bad. But they're not bad everywhere yet. It's a very patchy distribution, and it's centered in Calabasas. And Amy over here also gets a Hero Award for setting up all the traps at the Mountains Restoration Trust, Headwaters Corner, where sadly we found a lot of bad beetles. And it was a really kind of a sobering thing, because of course the MRT property is right up against Wild Woodlands, Wild Walnut Park. It's close to a lot of other, it's on that urban wildland fringe. So once again, we're looking at, a lot of risk. So this is the summary of where we found the bad beetles. So Calabasas totally got hammered. Parts of Chatsworth are totally hammered. A lot of these are either associated with mulch that was probably contaminated, or they're adjacent to green waste facilities where a lot of trees are brought for processing. A lot of those green waste facilities, even with tub grinders, use and sell that mulch, and it gets spread and distributed all over the place. The western oak bark beetle is definitely having a grand old population boom at the moment. So these are all the kind of biological issues associated with the loss of trees. But we thought it would be interesting and potentially helpful to understand what the ecosystem service values might be for these trees. And this is a calculation that's been refined over the past years, primarily by a researcher at UC Davis, Greg McPherson, who sadly just retired. But he's done amazing work in actually tying the amount of carbon storage, carbon sequestration, runoff from precipitation, and how much that actually translates into. And the city of Seattle, coincidentally, they did their calculations and realized it was cheaper to buy buffer zones along all of their river that runs through the city than it was to concrete the river and maintain it. We could take a lesson from that playbook for LA, just saying. But the thing about this is that it doesn't include the value of the trees for things like removing pollution or moderating temperature. You don't have to use your air conditioner because your tree's there. It doesn't have any value calculation for habitat or aesthetics. It doesn't include real estate values, and most importantly, it does not include what the costs are to remove a tree that's dead and dying and replace it, which we all know is significant. So these are very conservative numbers that I'm going to show you about how much trees are worth. And so if you look at it for an oak tree, it's about $300 per year of just carbon storage and sequestration, as well as runoff avoided. Most of these trees live over 100 years. So that's, you're getting to some big numbers here. And we estimated, based on our plots, that conservatively, with the NASA data on how many acres of oak woodlands that were pure stands, plus the oak woodlands that are mixed with other species of trees that National Park Service has calculated is about 11,000 acres. That gets us about 600,000 oak trees that we've got in the Santa Monica Mountains. And if we lose these trees, we're losing a lot of value. But what have we lost already? And thanks to the NASA, that heavy lifting that they did with their very sophisticated tools, we know that we've lost a lot so far. And the 60 million, and 45 million that we haven't lost yet is a lot of money. This is per year. This is not, and each tree lives 100 years. So think about that. This is enormous amount. So that gets us to the last and final part, which is what do we do? And you know me, I never like to leave an evening on a sour, sad note. So here are some things that we can positively do, each one of us, to make sure that we keep our forests going into the future. So the first thing is we can water our trees. If it doesn't rain, we need to make it rain for our trees. They cannot survive without water. And our water table has already dropped significantly. We're not going to be able to take water into our wildland trees. We darn well better do it for the areas where we are on the urban interface wildlife, where we still can do it. We can provide a buffer. We want to make sure that we're taking proper care of our tree roots and that we're not damaging them. This means that it's not time to, trench for your, footing for your power line to put in your Christmas tree lights. You just want to leave the roots alone as much as you can. And you want to have understory plantings that are compatible with the trees. Mulch is magic, and mulch is absolutely spectacular and wonderful, but we got to be careful where we get our mulch from now. I used to just say, get it from anywhere, it's great, use it all. But sadly, that is no longer the case because it could be contaminated with these bad beetles. So now you have to pay attention to where your mulch comes from. But still use it, please, because it does all kinds of incredible things. It's important to remember that the green leaves on the tree are what give it its energy. We do not feed trees. We give them nutrients and raw materials. Trees make their own food with their leaves. So if you prune out the leaves, you are taking away the tree's ability to take care of itself. So we should not be doing pruning unless it's absolutely necessary at this point in time. And, you know, if you have a tree that is infected, it's really important to take care of it properly, and that's really expensive. Right now, our best bet in this area is to the Lost Hills landfill because they have a big tub grinder, they grind everything up, and they use it as landfill cover. They are not selling it as mulch, at least not as last I heard. So this is a good way to do it. However, you have tipping fees now, so that makes taking the tree down even more expensive. The containment, I was cracking up. There was a video online. They're saying, oh yeah, solarize your stuff. You know, wrap it up really tightly in a plastic and put some hardware cloth over it, and that will contain it. Well, around here, it takes 6 to 9 months of full-on sun to begin to break down these beetles and kill them. And if you just use regular old plastic, the beetles just chew right through it and they're right out and about. So basically you're warming them up and sending them off. So I think it's probably not the best strategy in this area. Most importantly, you are all now ambassadors for not moving firewood ever, anywhere, anymore. We can't do that. And one of the things that I thought was really remarkable about this effort during the past year is that because of citizen volunteers who helped, we had so many eyes on the ground, and it's a really important element to keeping this going. And doing these beetle traps, as goofy as they were, was kind of fun, and it actually was really critical to giving us a lot of information. But in any case, we do need to come up with some other ways to monitor. And Bree has volunteered to help while she's working with us this year to see if we can set up an iNaturalist project where people can just use your phone, take a photo, and send it to us, and we can use the data from there. So we're working on that. Stay tuned. So just to summarize, I think it's really important that we make the connection between our health and well-being and what's happening in our environment around us. It is all connected. And the real antidote to all of this despair is to come out and help us plant trees. And I just would like to thank you all for coming tonight. I hope that you will continue to keep educating yourself on this. And I just would like to end with this thought from Sam Broder, who was speaking at this time about deforestation in Thailand. But I thought that this quote really kind of sums it all up, because the choices that we make on how we take care of our environment really come back. And trees are a key part of our environment. They are a key part of what keeps us alive and what keeps us happy and thriving. So the threat to our trees is real, it's scary, but we can do it. We can keep planting, and we will have trees for the future. Thank you.